Hi guys, welcome back. In the second part of the chapter 10 lecture, we're going to be talking about how eukaryotic RNA is processed before leaving the nucleus as mRNA. So the difference between pre-RNA and mRNA is that pre-mRNA is what exists in the nucleus before it leaves the nucleus so that translation can begin. Pre-mRNA is going to contain introns and exons, and basically these introns are going to be removed because they're not needed for the proteins that the cell desires. So after these introns are removed, the exons are going to be spliced together. So it's easy to think of it as a pattern because once you remove all the things that you don't need, everything that you do need, which is the exons, they're going to be spliced together, and this will create your strand of mRNA that you need. Now, when we talk about transferring RNA molecules, we can see that they serve as the interpreters during translation. Now, transfer RNA molecules are better known as tRNA, and this is what a tRNA is generally depicted as. So at the top, you can see that you have your amino acid, and this is your anticodon loop, which will attach itself to the mRNA codons. Now, there's two different functions of tRNA that you should take into account. The first one is that it can pick up the correct amino acid and then bring it to the assembly site for the peptide chain, and it can also recognize the right codons in mRNA using anticodons. So in this assembly process, you're going to have an E site, a P site, and an A site. Now, the E site is the easiest one to remember because E stands for exit. So basically, once you're done attaching the amino acid to the growing polypeptide chain, it's going to exit through the E site. Now, right next to it is the P site, and this is going to hold the growing polypeptide chain, as you can see here. And then lastly, the A site holds the next polypeptide that's in line to be attached. So the amino acid is going to start off here, and then once it's inserted onto the growing chain, it will exit through here. Now, elongation is the process that adds amino acids to the polypeptide chain until it reaches the stop codon. So basically, in the process of elongation, the codon is going to be at the A site, and then it's going to move over to the P site so that the chain can elongate itself. This is the growing chain in this picture, and then once it's done being attached, it will exit through the E site. Now, as we discussed before, the process of transcription and translation are very rapid processes, and this can all give rise to the presence of mutations. We're going to be looking at three different types of mutations. The first is a silent mutation, and basically this means that there's no harmful effect because the same amino acid is still present in the chain. And the reason this is possible is because there can be several codons that all code for the same amino acid. So say you have a mutation in the last codon of in the last base of your triplet, it doesn't always mean that you're going to now have a stop codon present or a completely new amino acid. So whenever this happens, it's called a silent mutation. But in a missense mutation, it means that the change in base pair or the change in base is actually going to result in a different amino acid. So whenever you have a chain of amino acids that all code for a protein, if you have an alteration in one of the amino acids, it's not always going to result in the same protein being made. So that's why this can be problematic for a cell. Now, in a nonsense mutation, it means that the actual chain of amino acid is going to be inhibited or stopped prematurely because the chain is going to result in a stop codon. Now, frameshift mutations are often seen as the most harmful because you can have an insertion or a deletion of a base pair that can throw off the entire frame of the bases that are being transcribed, and this can lead to a chain of amino acids that is completely different than what was originally desired. Now in this picture, we can see here that this is the normal strand of amino acids that we need. Now in a silent mutation, you can see that we have an error here because this G is actually supposed to be an A in the normal scenario. However, both of these code for the same amino acid, so you don't have any problem. Whereas in this missense mutation, the A is now a C. So this leads to a completely new amino acid, and this will lead to changes in the actual protein that is going to be produced. Now, in a nonsense mutation, you can see that the chain of amino acids stops elongating prematurely because this alteration or mutation will lead to the presence of a stop codon, which means that no more amino acids can be added to this strain. Now, in the frameshift mutation, what you're going to have is that of this normal strand, say that we delete this A right here, or say we delete this T, then that means that this codon is going to be ATC, and the rest of the ones following after this one will all be changed as well. So now you have a completely different strand of amino acids than you originally had desired. Now, getting into the genetics of viruses and bacteria, we're going to be talking about viral structure and viral reproduction. So basically, the structure of a virus is that there is DNA or RNA, which is going to be the genetic material of a virus, and it's going to be encapsulated in a protein coat. Now, in viral reproduction, there are two different phases that we're going to be discussing. The first one is the lytic phase, and the second is the lysogenic phase. And the best way to understand these two phases is by looking at a diagram. So we're going to be looking at this one right here. Now, basically with lytic phases, the bacterial cell is going to be lysed or destroyed after the immediate replication of the viral material. So as we can see here, as soon as the cell is lysed, you're going to have the phage progeny basically escaping the destroyed cell, and now this progeny is free to go around the organism and infect new cells. So this way, you can have infection spread a lot faster, whereas in the lysogenic cycle, it does not always ultimately result in immediate lysing of the cell. So that's what makes it a bit different. Now, the phages that undergo lysogeny are known as temperate phages, and their viral genome integrates itself with the host DNA, and it will replicate along with it almost harmlessly, and it might even become established as a plasmid in some instances. Now, this case is unique because the virus can actually remain dormant until the host conditions will deteriorate, and this can be due to a variety of reasons. One of them could be the depletion of nutrients. And then the prophages are going to become active at this point. Now since they're at this point, they'll be able to initiate the reproductive cycle, and this will result in the lysis of the host cell. So as the lysogenic cycle allows the host cell to continue to survive and reproduce, the virus is reproduced in all of the cell's offspring. Now there are three different ways in which bacteria can transfer DNA. The first one is through bacterial chromosomes, which we have here. The second is through transduction, which is the transfer of bacterial genes through a phage, and this can be seen here. And we also have transformation, which is whenever you take in foreign DNA from the environment. So what this would look like is that once the DNA is inserted into the cell, it's going to integrate itself with the DNA of the host cell itself. So now every single time this cell is going to reproduce and make new cells, the genetic material is going to incorporate the actual genome of the virus. And lastly, in conjugation, you have exchange of DNA between two bacterial cells, which we can see here. So now we have officially finished talking about Chapter 10 and discussing the process of translation and transcription in cells. If you guys have any other questions, you can send an email to brhssciencehonors@gmail.com, or you can also send us a text message through Remind. And we will see you guys in our next video.